Okay, so now let's practice a little bit with the Henderson-Hasselbalch equation. We've talked about generally what pH is, how to calculate pH, what pKa is, what it means about an acid, and how to use pH and pKa to get information about concentration. Now let's talk about the idea of a buffer. Buffers are just a solution that mixes a weak acid and its conjugate base together. And in fact, in this last example, testing out the Henderson-Hasselbalch equation, this was a buffer. We had a weak acid, acetic acid, and we had its conjugate base, sodium acetate, or the acetate ion. That is a buffer. We mix the two together. Buffers are very resistant to changes in pH. Now, when you make a buffer, at the pH equal to its pKa, you know that there is a 50-50 mixture of the acid and the ion, the conjugate acid, essentially. So at this point, we know that it has dissociated halfway, 50-50. So half of it is this HA, and half of it is this A minus. And at this point, it has the best buffering capacity. You can actually see that graphically when we take a particular acid and we keep changing the pH little by little and looking at, as we add more of a counter ion, as we add more base, for example, how the pH responds. So here, this is an example of what we just saw, acetic acid, and again, its pKa is 4.76. So let's say you just have pure acetic acid. That's the acid form. That means to begin with, we actually have concentration of HA that is very high. Right? We just have HA. We have not dissociated it into a counter ion. So initially, we have HA that is very high. And as you add a little bit of OH at a time, and this is just an experiment to show buffering capability, but understand that if you added a little bit, if you titrate a little bit of OH at a time, what you would see is that around a pH equal to its pKa, so around, in this case, 4.76, we're going to have a good balance of the HA. In fact, have a 50-50 relation of HA and A minus. Why do we want this? Why does this make for a good buffer area? At this point, when you have a pretty equivalent amount of HA and A minus in the system, you can easily intake any OH minuses or H pluses around that level and quickly convert between the two. So it allows to quickly deal with a pH change, quickly deal with some H plus or some OH minus that has been added to the system. And that's why right around here, even though we're adding more OH, seems to kind of flatten out. The pH doesn't really change that much. So that's right around good buffering capacity, around a pH of 4.76. Now, of course, you can eventually knock it out of that range and keep adding OH. We could even do the opposite in this case if we started with a bunch of this CH3COO minus, that's A minus in this case. Remember, this is the pH. If we did the opposite, we went backwards and we added a bunch of H plus and we eventually lowered the pH, you still see this flattening curve, the buffering region. So again, the take home here is that the buffering region of a weak acid is going to be at its pKa. And in general, a plus or minus 1 range of its pKa. Now, because each acid dissociates differently, each acid has a different pKa, a different constant. That means that different acid conjugate-based pairs have different buffering zones. So the buffering capacity is going to be based on that particular set of ions. So here again are some examples of different ion pairs, the acid and its conjugate base, the pKa of that particular pair, and so the buffering region for that particular acid-base pair. And again, remember, this is a region, so the best spot is when pKa equals the pH, but the general buffering region has a plus or minus of 1, where it still acts as a pretty good buffer. Now, buffers biologically are very important. Within a organism, they're useful for maintenance of intracellular pH. This is vital to cells. That's because certain reactions can only occur at optimum pHs. And also, solubility of different molecules can be dependent on the pH. It's even especially important for something like our ability to breathe. That's because the equilibrium between CO2 gas that is dissolved is dependent on the pH of our blood. Buffering systems are mainly based on molecules like phosphate, bicarbonate, which we see in blood, and histidine. This is another small molecule also found in the body and also very important for efficient buffering of pH. Now, outside of living organisms, we still have other buffering systems. And in fact, as biochemists, we rely on these other molecular buffers to stabilize some biological components. When we're working with proteins and different biomolecules outside of a living organism, we still have to keep them in a stable pH. And to do that, we make buffers. And in fact, we'll be talking about this a lot for those of you who are in labs making buffers. And hopefully when you get Biochem 2, we'll actually practice some of that in person. We'll be actually making the buffers and showing how you calculate the pH, that are what we're practicing today, and then doing it. So some of these buffers you'll see a lot written about. HEPES, that is a very, very common one. You see a lot. PIPES and CHPES, these are all buffers you don't find in biological organisms, but we use often to make buffers stabilize biochemicals. Okay, so let's do another practice thinking about buffers, and again, the Henderson-Hasselbalch equation. I think we're going to be using this equation a lot. Okay, this one is more medically directed. Aspirin is a weak acid, and its pKa is 3.5. It's absorbed into the blood through cells lining the stomach and the small intestine when it's passing through. And that absorption, its ability to pass from inside your stomach or inside your small intestine into the bloodstream, requires it to go through a plasma membrane. Now, the thing about plasma membranes is something that is charged or highly polar does not pass through it very well. That's because plasma membranes are made of physics. But things that are neutral or hydrophobic pass very easily. The question here is, if the pH of the stomach's content is 1.5, and the pH of the contents of the small intestine is 6, is aspirin absorbed more into the bloodstream when it's in the stomach, or when it is in the small intestine? Okay, so first, let's go back to that first paragraph. Aspirin generally is going to be a weak acid. That means it will dissociate, and it is going to dissociate to a 50-50 point, right, and pH around 6.5. We'll get back to that in a second. Let's write our equation for weak acid. HA goes to H plus A minus. This is the acid as its main form with its proton attached. And it is dissociated to give away an H plus and made its conjugate base. So aspirin in its pill form is going in as this. Going in as a weak acid HA. And it will eventually dissociate into H plus and A minus. So which one of these forms, first of all, is going to be better at passing through the plasma membrane? The HA, the acid form, or the conjugate base form, the A minus? Just look at this general equation. You don't even need to see the structure of aspirin. You can just understand that the conjugate base form is going to be more charged / polar. It has a negative charge on it. Whereas when aspirin is in its conjugate acid form with the proton still attached, it's a little bit more neutral. So that means we want to have, based on what we're seeing here, something that is more neutral, so more HA. Okay, now let's get back to this, looking at the pH content of two different areas, the stomach or the small intestine, and how is that going to change this concentration of HA to A minus? Well, we can figure that out using the Henderson-Hasselbalch equation. Remember again that the Henderson-Hasselbalch equation says that pH equals PAA plus the log HA A minus over HA. Okay, so in something like the stomach, our pH is 1.5. The pKa for aspirin is 3.5. And we don't know the concentrations of these two, but we can see how different pHs will affect the concentration of the A minus over the HA. Now, in the small intestine, we have 6 as our pH, pKa of and it's the same. And we'll see how that environment affects this ratio of the conjugate base and the acid. Doing a little math here, we're going to rearrange the gamma system. Stomach, and this is the small intestine. In our stomach, if we were to rearrange this, let's subtract out this number. Get 1.5 minus 3.5, which will give us a negative 2.5 equals concentration of A minus over concentration of A. Similarly here, I'm going to do 6 minus 3.5, and that gives us 2.5 of this equals log of concentration of A minus over concentration of A. Okay, so lastly, how do we get rid of that log? Remember, to get rid of the log, we have to raise both sides to the power of 10. So that means 10 to the negative 2, and we get rid of the log over here. And on this one, it would be 10 to the 2.5. And this those are going to be equal to this ratio. Well, 10 to the negative 2 is the same as 0.01, and 10 to the 2.5 is the same as 316. So we have a number, 0.01 is the ratio of this A minus to HA. And down here in the small intestine, 316 is the ratio of A minus to HA. Now, just looking at this, I can tell that this is a small number and this is a big number. So that must mean that if I look at this ratio for a small number, the A minus on the top has to be small, and thus the HA on the bottom has to be big. Oppositely, if I have a big number, that means that the A minus on the top is big. And this has to be small. Again, just to get a large number larger than 1, this ratio has to be true. And to get a small number, this ratio has to be true. Now remember, which one is better for passing through a plasma membrane? Something that is more neutral, so that means the HA, not the charged one. That means this situation in the stomach is the better situation because there is more HA. The ratio of HA to A minus is bigger. So aspirin will be absorbed more. Again, buffers themselves are very important, as noted previously. There are tons of proteins with certain amino acids in their functional groups that are weak acids and weak bases. And these are going to be buffered in different ways based on the weak acid and weak bases present in the aqueous environment itself. If you're still not sure why buffers are important and why they matter, I've linked something here that goes really into depth in each of the organelle systems and organs and how they utilize phosphorus in the body. One of the main ones I'm going to focus on is buffering in blood. The blood pH is controlled by the amount of carbon dioxide and bicarbonate in our blood buffering system. And this is called the carbon dioxide bicarbonate buffer system. It can be shown in this equation. Here we see that CO2 as a gas will mix with water within our blood to form carbonate. That's this molecule, carbonate. And ultimately, this carbonate will break down into a H plus and this HCO3 minus. This is called bicarbonate. During hypoventilation, the concentration of CO2 in the lungs and in the arterial blood will increase. That means hypoventilation is not getting enough air, so CO2 starts to increase. In this situation, we would drive our equilibrium to the right, and it would raise pH plus. So I have a lot of CO2. That means my system will start trying to drive this way, right? We have too many reactants to start trying to mix one up. That means it's going to drive up the amount of H plus and ultimately make the environment more acidic. So the pH will become low, it will become an acidic environment. So when we have too much CO2, our blood becomes more acidic. In the case of hyperventilation, the concentration of the CO2 in the lungs and the arterial blood is going to fall. This is in a case where we are taking in lots of oxygen and we don't have a lot of CO2. Hyperventilating, right? Breathing fast. That's lots of oxygen in, not a lot of CO2 out. So our concentrations of CO2 are going low, and this will drive the equilibrium equation to be less towards making CO2. We have lots of oxygen, we're going to make more CO2. This requires that we use H plus, and thus it will increase pH. We're going to be having less H plus around. We're going to have a basic situation that increases the pH. And again, in a hypoventilation system, when we have too much CO2 present, we're not getting enough oxygen, the blood will become more acidic. It'll have a higher pH. And in a hyperventilation situation, where we don't have that much CO2, so we're actually intaking more oxygen, this will drive our blood to become more acidic, lowering the pH. So let's look at an example of, again, using the Henderson-Hasselbalch equation, but in relation to a blood plasma sample. Here, I want to try to calculate the pH of some blood plasma sample. If we know the total amount of CO2 concentration and the total amount of bicarbonate concentration. We also know the relevant pKa. So for blood, again, this is important to know this distinction. Our blood buffering system, again, is this kind of complex equation where we have CO2 that goes to what we're calling carbonate. And again, I will write that out so you see what that is. H2CO3. And then this is what is ultimately buffering to form bicarbonate and H plus. Bicarbonate looks like this, H2O. So really, these two are the main ones doing the buffering, carbonate and bicarbonate. But so you're aware, they come from CO2 being dissolved in the blood. So the amount of carbonate and bicarbonate that is here is coming from CO2. So I'll write this. This is an important thing to know. The amount of CO2 is equal to the amount of the carbonate plus the amount of the bicarbonate. And again, that's just where this buffering system is coming from. CO2 is going to make the carbonate, which is then in relation, it's actually the acid group pair with bicarbonate. The CO2 is the concentration of CO2 is the addition of these. Okay, so again, we know what the acid base pair are. That is the carbonate. We call this HA, the acid. And its conjugate base is the bicarbonate. Okay, now that we have all of those pieces of the puzzle, let's use the Henderson-Hasselbalch equation. pH, which is what we want to solve for, equals the pKa plus the log of the conjugate base over conjugate acid. Okay, so here we know our pKa. It is 6.1. Now remember, in this equation, our conjugate acid and conjugate base are carbonate and bicarbonate. But we have information about the total CO2 concentration and the bicarbonate concentration. Okay, well, we know bicarbonate is here. But how do we know how much carbonate there is in the system? Well, remember, I told you that carbonate and bicarbonate just come from CO2. So if you knew the total amount of CO2, you can calculate your carbonate and bicarbonate. So total amount of CO2, 26.9. That's equal to the amount of carbonate present. That's what we need to find. Plus the amount of bicarbonate present, which we know. 5.6. So rearrange this to be 26.9 minus 26.6. We get 1.3 equals the amount of carbonate. Okay, so that means the amount of carbonate here is going to be 1.3. So now we have all of our pieces filled in. Let's quickly calculate our pH then. So in this case, let's first do the 25.6 divided by 1.3. That gives you 19.69. Now we have pH equals 6.1 plus the log of 19.69. If I calculate the log of 19.69, then I get 1.29. That means our pH is going to be 6.1 plus 1.29, which equals 7.4. So again, our pH of this particular blood sample is 7.4. That is right around a pretty biologically standard pH, which means that the amount of CO2 in this blood sample is good amount. It is not a case where we have hyperventilation, hypoventilation. Okay, so in summary here, in the whole of this chapter, we talked about water, and we talked about the nature of water's intermolecular forces. We also talked about properties and structure of water. And here we're really focused on the behavior of weak acids and bases in water, how to talk about them using pKa and pH, what those numbers mean, and how to calculate between concentration, pH, and A. We also talked about ways water participates in these reactions via a buffering system. We talked a lot about blood buffering and other buffering systems. And again, how to get information about that buffering system.